Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Kilty. Hello, everyone. Welcome back to another episode of our Jack Westin MCAT Podcast with your hosts, Mike and Molly. In this episode, we're going to be diving in to a really interesting topic, which has kind of been developing throughout the past couple episodes, right? We really started with the brain and then talked about a couple key functions of the brain, vital functions. We talked about movement, and then we started talking about waves and physics, and we talked about sound waves and hearing, and then now light waves and optics. In this episode, we get to figure out, okay, well, how do we actually sense and perceive light? Okay, so coming from our optics episode, which was our last one, we're going to be talking all about the eyes and the occipital lobe. Basically, how do we sense and perceive light, which will be pretty cool.
It's super cool because we're kind of like on this track of like we've got these natural phenomenon happening around us, but our brain has to interpret these things. We talked about like in the last time we talked about like the sensation, we talked about hearing, right? We talked about how our ears, this really cool structure, turns these sound waves into electrical signals in the brain. We're going to be doing the exact same thing except with light. And it's very cool because I think when we're discussing about these things, there are similarities in terms of like turning waves into electrical signals, but with some very key differences as well. Like actually the how it does that is pretty different. But it's very cool how we do that, and how all the structures involved and how unique these structures are, I think, in the body, like that these cells, are very unique to the eyes. You don't see them anywhere else. So it's very cool. And it does really build off what we talked about last time. But when we really delved into like what light actually is and these different ways that the MCAT can test us, especially when it talks about like reflection and refraction. And we're going to see not even just a lot of that here when it comes to the eyes. And that's actually, I think, where if we were to map out where we're kind of like we're going to be talking today, we're going to be talking about how first light enters the eye, some cool things that happens there when it enters the eye, once it gets to the eye, where does it need to go to actually be turned into a signal, and once it's actually turned into a signal, where it's going to end up in the brain. So I'm super excited to dive in.
Yes. And this content is something that in at least certain portions of it could really be covered in any science section. Right? I would say vast majority you're probably going to see in Psych Soc, but you can absolutely see some of this stuff in Chem Phys or Biochem, right? Like it is fair game. It may not be the entirety of what we're talking about, but if we're talking about light and refraction in the lens, that's just straight up physics, right? Like you could get calculation-based questions about that. You could be given the focal length and the object distance and calculate the image distance of an image with the eye and the actual, the cornea and the lens. Like this could be in Chem Phys. It could certainly be in Biochem as well. And again, probably the most in-depth with Psych Soc, but whenever you have a topic that really can straddle any science section, it's really important to understand it very well because it's very likely to show up. So this would be cool. And you're right, we're going to go through a couple different like layers, so to speak, or major phases of this process, right? The first thing is the actual light. This is more physics related, right? Light is entering the eye and then it's going to strike objects or structures within the eye that have very different indices of refraction, which is going to cause light to bend, right? That's the cornea, the lens, which we'll talk about. That's all very optics. Okay, we have not had any sensation or perception yet. It's just literally light is entering the eye, right? It's traveling through the eye to the retina, which is on the back of the eyeball, right? And then that is where we actually have our photoreceptors, which are going to convert light energy into a neural impulse. Okay, and then that is then, right? So that's like transduction, maybe you could call it, right? And then we've got to travel to the region of the brain within the occipital lobe that's actually going to process and interpret what is being sensed by that retina. Okay, so we've got a couple major phases, and it's going to be fun to go through each one, right? So let's start with the optics of it all. Okay, so the actual light entering the eye. Okay, now in our previous episodes, we've talked a lot about how light travels through a vacuum or air or liquids or solids, right? Um, and of course, most of the time, unless you are a scuba diver or your, eyes are open under the surface of the pool or the ocean, you're probably seeing and sensing light that's coming from the air, right? It's going to the cornea, which is going to be the major actual source of refraction, right? So the cornea is going to be the portion of the eye that light is traveling through first. And it's actually got a very different index of refraction. I think, and I'm pulling this from memory of a passage, I think it's like 1.36 or something like that.
That
Very
that
different
sounds
from one.
about that sounds about right. Like kind of similar to water because it is mostly made up of like water and fluid. So
Yeah.
there's a little bit more there. So I believe because water is like 1.33. So like it does sound like right. That'd be just slightly above water, 100%.
Yeah.
So,
I'm
mm,
literally pulling it from memory. Keep in mind, you don't have to memorize any, you don't have to memorize the index of refraction of the cornea. It will always be given to you. Okay. So
mm.
no worries. But it's very different from one, right? And what's going to happen, Mike, when you travel from a substance or a medium with a low index of refraction to one with the high index of refraction?
Well, that means the speed is going to change. And so therefore
Mhm.
it will bend. You're going to a
Mhm.
new medium, so it is going to bend. And we talked about before that the possibility when you go from like high to low of like total internal reflection. But here,
Mhm.
right, we're going from the air into a cornea. So which is low to high. And in that case, no total internal reflection occurs here. It's all refraction. It's going to go into that medium. It's going to change speed because we know the speed of a wave depends entirely on the medium that it travels. And so since it's a new medium, the speed will change and therefore it will bend. So yeah, it's gonna bend.
Yes, totally. Right. And it tends to move slower, right, as it goes
Mm,
in, which is going to cause that bending. And I think most people, when they think of light entering the eye, right, and they know that light is changing as it's going through your eye, right, that
mm,
most people think about that. What I find is most people think the lens is mostly responsible for this.
mm,
And the cool part is actually the lens is more of a fine tuning with how light is going to bend. Okay, so
mm,
really it's the cornea that does the majority of the refraction or the bending of light that's entering the eye, which is pretty interesting, right? Again, it's got a very different index of refraction. And fun fact, if anyone, any of our listeners out there also need contacts or glasses, like I do, Mike doesn't. He's got perfect 20/20
Oh,
vision. Good on
stop.
Mike, right? We're all
Stop.
jealous.
Oh, stop.
For those of us who do need some sort of corrective eyewear, you may have heard of LASIK before. And this oftentimes, again, people think it's reshaping the lens, but it's actually using laser technology to reshape the cornea
Mhm,
to adjust refraction because that is the major refractor, right, in the eye. It's not the lens.
mhm,
Although, of course, as light is bent from the cornea, it will, of course, pass through our lens, which most of us are a little more comfortable with and used to talking about, perhaps. But this is really just a very, very minute, more
mhm,
adjustment-based
mhm,
refraction. Really just helping us take into account, well, how far is this thing away from me? How do I
mhm.
focus it so it goes straight on the retina?
Right.
Right? But Mike, how does it do this? Right? Like how
Right.
does it know how much to bend light, right? In
Right.
that adaptive version.
And that is the kind of cool part where the lens is the adjuster, right? Where
Mhm.
when a lot of times we've been kind of mentioning a lot where it's like, oh, we need to bend the light. Need to bend the light. A lot of people are like, why do I need to bend this light? Why can't I
True.
just have no lens, no cornea? Why can't I just let the light in? It's because there's so many light rays hitting your eye. Like I'm like looking around, there's so many light rays in my eye. But yet, even though there's so many light rays, I can see crisp images. It's
Yeah, we
because
get it. You have perfect eyesight,
yeah,
Mike.
I know. I like to flex a lot about my favorite eyes. Right. Of course. 2020. Yep, we just got my eyes tested. But anyway, but all of these light rays are hitting my eye, but I see a crisp image, or we all see crisp images if we do it correct, if we eyes are anyway. Because sorry, well, I like to encompass my non 2020 friends like Molly. Because these light rays are getting focused. Now that's the kind of key where our rays, when they hit the eye, need to be focused on an area of the eye known as the retina, which is the back of their eye. The kind of this back wall of the eye is covered in these little cells known as photoreceptor cells. We have our cones and our rods. And it's by focusing all on a specific point on the retina, known as like the fovea centralis or the fovea, where there's a lot of these cones, we can see things clearly. Because if we didn't have any bending of light, right, they would just be spread over the retina. There'd be no focused image. Everything would be blurry. And that's why those who need glasses, when you don't have your glasses or corrective lenses, everything's blurry because they're not focused on that retina, on that, fovea in the center there. And so we need things like the cornea, things like the lens to do the focusing. Again, the cornea does the majority of the bending, right? And that's fixed, right? It's like the cornea doesn't change the bending any amount. Like it's fixed, it's always bending it the same amount. It's the lens that's fine-tuning that adjustment because, like you said, we're seeing things close up, we're seeing things far away where the light's going to be coming at slightly different angles each time. So the lens needs to adjust so that every single time it adjusts exactly on the retina so that we can actually see a crisp image. And it's when you're not seeing a crisp image, there's something's going on where it's not quite focusing on the retina. And that's where we're talking about like things like myopia and hyperopia. And so since I can't really relate to those sorts of things, I'll let Molly talk about these things.
Oh my God, I'm deceased. Yes, I do have terrible eyesight. Thank you. Yeah, but also to your point, I love thinking about the lens. If anyone, by the way, is interested in cameras or has had like a DSLR or anything like that, you can sometimes see if you're adjusting the zoom on your camera to change the focus depending on how far away your object is, you can see the lens moving around, right? Especially in a DSLR with like a big giant extra lens that you add to the camera, right? You might make it longer or shorter, right? Move that lens around, change its shape. It's pretty cool, right? And it's basically what's going on in your eyes. There's a lot of similarities between cameras and your eyeballs, which is super cool. So it can also kind of help to externalize to something that you can see. And funny enough, if you're seeing the camera, you're also practicing the
Oh my God.
actual
This is getting like
sense of sight,
meta.
which this all applies to. So, love that. If you are like me and you don't have perfect eyesight, you might be thinking, I wish I could just adjust my zoom and adjust my focus, with the touch of a button, which of course you can't. However, you can use things like contacts and glasses. Okay, so if you are someone who wakes up and you don't see the world perfectly crisp, nowadays, right, you have the ability to fix that in a variety of different ways. We've already talked about LASIX, which is surgery, right? But also, if you can't see far away or you can't see up close, you can fix it pretty easily. Okay, so these conditions of seeing blurry, vision are put into two camps, either myopia or hyperopia. Okay, now I always remember this based on myself. And if you are in your, younger ages, right? Like typically between, birth and like maybe 30 or 40, this is more common to have myopia. I have myopia. I'm what's called nearsighted, meaning I can see near, but I struggle to see clearly objects that are more farther away. Okay. If you are far-sighted, it means you can see things that are far, but it's harder to focus on things that are near. Okay. It is more common for younger individuals to have myopia than hyperopia. That being said, most of us as we age will develop farsightedness, right? It will be harder to see things closer up. That's actually due to corneal changes more than anything else as we age, but just so you know. So myopia or being nearsighted is essentially this issue where light is being focused too close to the lens, meaning it hasn't gone back far enough to actually focus on the retina. Okay, so the perfect little image that's being displayed is in front of the retina instead of on the retina to provide clear vision, right? So this is a problem. We're focusing too early, okay? How might we want to fix this, right? Well, if our cornea and lens are not refracting exactly the way that we need to, right? It's almost like we're refracting too much, right? We're bending too much that it's focusing before we want it. We're going to want to diverge those light rays a little bit, spread them out so that when they go through the retina, or sorry, they go through the cornea and the lens, they focus perfectly on the retina. So if you have myopia like me, or you're nearsighted, you should go for a diverging lens. Okay. And again, this is talking about glasses or contacts, right? They are lenses that you put in front of your cornea and your lens to adjust that focusing so that it is going perfectly on the retina and giving you a very clear field of vision. Okay.
Yeah.
On the flip side, if you have hyperopia, meaning you're farsighted, okay, your image is actually being focused behind the retina instead of in front of it. Okay. So it's that perfect crisp image is too far back. In that case, we want to bring it forward. We want that light to converge earlier. So, as I kind of alluded to with that
Mhm.
choice of words, you want a converging lens, right? That's going to allow light to focus closer to the lens itself, which then will allow it to focus clearly on the retina. Again, if the image produced by your complex optic system, right, of your cornea and the lens and possibly some contacts or glasses, if it is perfectly focused on the retina, that is when you have clear vision.
Mhm.
So if you, just to summarize, if you are focusing before the retina, like myopia, nearsightedness, like me, and probably like most of us, if you wear contacts or glasses, you need a diverging lens. If we are talking about hyperopia or farsightedness, we're focusing after or behind the retina, you want a converging lens to bring it up. I feel
Yeah,
like
and I
that's the
think
most clear application to our optics discussion of last episode.
And I think it's so cool because it's something that a lot of, like, honestly, I would say like a majority of people who are taking like the MCAT, like a lot of people have glasses. A lot of people have
So
glasses,
common.
and very, very common. And this is just one area where your lived experience can actually like impact like how well you understand things. I mean, we talk about that all the time in psych soc, but it's also a great application where a lot of people who are nearsighted or myopia, when you ask them the prescription, they're like, "Oh yeah, I'm like negative X." Oh, wait a minute, negative. Reminds you
Mhm.
it's, oh, what kind of lens? Oh, it must be some sort of a diverging lens. Because this is something we, kind of a review of last episode, where we talked about, oh, some of the times you have diverging and converging lens. Well, if it's a diverging lens, the focal length or the focal point will be negative. And again, you might be like, negative focal point. Well, by you saying you have a negative prescription, that's you admitting that you have a negative focal point. So that's kind of like a great memory that I like use with students where it's like, hey, if you've got a negative prescription, that means you have a diverging lines because it is a negative focal point. Whereas, hey, if you were farsighted, which is, like you said, like far less common, if people are like, yeah, I have positive prescriptions, I'm like, first, double check because it's very uncommon. But if you do, right, you truly do, yeah, that means you have a positive. That means you have a converging lens, which is also a great reminder that focal length or if the sign of the focal length isn't always positive, which a lot of people kind of mistake.
Yeah. And
Mm,
it's not that hyperopia is uncommon. It's just not as common when you're young. It's something that
mm,
you tend to develop as you age.
mm,
Fun fact though, if you're ever like checking out at CVS or Target or Walgreens or something and you see the little tower of reading glasses, those will
mm.
all have positive focal or
Because
positive
mhm,
prescriptions.
target
Like
audience.
+1 or +1.25. It's because they're meant to give things that are
Yeah.
closer to you. So fun
Mhm,
fact, kind of
mhm.
cool. You'll see this next time you're again like in a pharmacy or a drugstore or something like that, which is
Yeah.
kind of fun.
But like so say now, like let's say, you had hyperopia, myopia, but now you get corrective lenses. Wonderful, great. It's corrected where it's focusing. It is now focusing that light at the retina. Big win. Story's not done yet. Because the light is still a wave. It hasn't been turned into a signal because something we kind of hit on kind of consistently in this kind of last few episodes is for us to ever feel something or perceive or sense something, it first needs to be turned into an electrical signal. If it is
Yeah.
not turned into an electrical signal, our brain will not be able to sense it. And so that's the kind of key with all of our sensations. It's turning that wave into an electrical signal, and that's the role of the retina. We've now focused that light on the retina. Well, now this is where the physics becomes biology, where our yeah, I know you like that one.
I like
Where
that.
this is where we're kind of like we talked about before. This is a section that like can really be tested in any of the science sections. Before we were very much like, yeah, lenses, optics, that's physics, right? But now we're like, okay, now we're talking about some cells and some things in some cells and some signal transduction that can be very related to biology. And so when we get onto the retina, we zoom in, we actually have multiple layers of cells, multiple layers. The first layer I want to discuss is the photoreceptors. Specifically, there's two kinds of photoreceptors, the rods and the cones. And what's the most interesting? I always find this weird about vision. Like if I were designing the eye, I wouldn't have designed it this way. But the rods and cones are in the back of the retina. Like you've got multiple layers, right? You've got, the ganglion cells, the bipolar cells. We'll talk about those in a second. And the light has to pass through those ones to get to the photoreceptors in the back, right? Which I always found kind of strange. I'm like, wouldn't the thing that you're trying to target be in the front of the retina? Anyway, weird side story. But either way, so you've got multiple layers. It's in the back of the retina, these kind of photoreceptors. Now, when it comes to rods and cones, they do have very, very different functions and different localizations on the eye. It's not like the eye is just an even distribution. Like, oh, and for every rod, there's a cone on the back of the eye and they're all evenly distributed. No, no, no. Rods have a very specific function. Rods are very much focused on low light, right? Just black and white, and are not associated with color whatsoever. Rods are no color, low light, whereas cones are very focused on bright light, colors, high acuity vision. And so when we are focusing on something, we want that light focused on cones, right? And that's why if we were to look at the distribution of rods and cones on your eye, there's one area of the eye that has essentially all the cones in your eye. Like all the cones are essentially in just one area, and everywhere around that area are rods, right? And this area of the eye with a high concentration of cones is known as the fovea, or you might see that as the fovea centralis. Right? Like if you looked at an eye, I think it's called a fundoscopy, when you actually like look at the eye, or you do like a scan, it's that center, that point, that's where all the cones are. And when you're focusing on something, what you're doing is focusing the light on that point with all the cones, because this is where you're going to be high focus. You can see the details. And again, light that like doesn't hit the fovea, it's not like you don't, it's not like blackness, right? That's where your peripheral vision is going to be, where it's more less about details, more like, okay, is there light coming from there or not? But yeah, it's that center area, that's where all the cones are. So I always think cones see color, right? That's how like cones are color, rods are not color. Like that's how I kind of remember the difference between the two.
Yeah. I always did fovea and focus.
Ah,
You
I
focus
like
on the
that.
fovea.
Right, right.
Yeah, we always have fun conversations of like, oh my God, wait, that works too. I always learned it this way.
That also works. But
Like, oh,
so,
hey, wow.
oh, wow, that's
Multiple
cool.
ways to learn. Who knew?
We love that. But now we focus this light on the fovea, let's say. Now let's think about what happens, because the common thing is like, oh, that means, oh, those cells turn on, they become active, and now we get it. Nope, stop right there. Let's hold it back. Because you would think so. But this is what I mean when I was like, if I were designing the eye, I wouldn't have done this way, but evolutionary biology does its thing, and I'm sure Molly, well, I'm sure there is a reason for this. But it's actually very interesting where your rods and cones are actually inhibitory. And what that means is like when there is no light, they're constantly what we say depolarized. And they're constantly releasing a certain neurotransmitter. That neurotransmitter is glutamate, right? They're constantly releasing glutamate to another type of cell known as a bipolar cell. And what that glutamate is doing, it's inhibiting that bipolar cell to say, hey, there's no light happening. Turn off, turn off, turn off. So it's the cones and rods, they're actively producing an inhibitory neurotransmitter. Now, I'm saying that about glutamate. And now you might be thinking, but Mike, isn't glutamate an excitatory neurotransmitter? That's kind of like in the brain. In the eyeball, it's inhibiting, right? I know that's a no. I'm sure somebody's thinking that. Again, I didn't design this, I promise. Don't shoot the messenger. But glutamate in the brain, very excitatory. In the eye, though, it's inhibiting the bipolar cells, and these rods and cones are constantly depolarized and releasing it. But therefore, what light does is it goes to the eye and says, hey, turn off, hyperpolarize. And what it does is it actually inhibits the rods and the cones. When light hits rods and cones, it essentially hyperpolarizes them and causes them to stop releasing neurotransmitter. And it's by not releasing an inhibitory neurotransmitter that actually activates the next cell. And so it's so counterintuitive. I know. I'm really if you didn't notice it, I promise I didn't make the rules. But it's by hitting these rods and cones, we're turning them off essentially and allowing the bipolar cells, the next cell down the line, to turn on and actually send a signal. I just found that always cool, and I found it confusing when I know, so that's why I'm like I want to really make that very clear for anyone listening right now.
Yeah. Yeah.
Mm.
I mean, I want to jump in, but you got to keep going. You got to finish this up.
Sure. So
Take us to the optic nerve because
sure.
I know you care so much about this.
Because again, I'm like, who designed this thing? And I'm like, oh, evolutionary biology did. Thank you. But so then, okay, so once you have, okay, cones and rods, they're not inhibiting it anymore. Great. So now you have your bipolar cells. But now, because the bipolar cells, they were like being inhibited the whole time. They're like, I'm kind of sleeping because I've got all this glutamate on me. But now they're kind of just like chilling. All right. But now there's no more glutamate anymore. So now they're like, oh, hey, okay, that must mean there's light because there's light turning off these cones and rods. So now the bipolar cells are like, signal now. I'm going to activate now. And they send them to the next line of cells, which is the ganglion cells. And it's kind of interesting because the signal kind of goes forward. You went to the furthermost back cells, and now you're going forward, kind of deal, towards the front of the eye, which I always thought was, again, strange, but okay. I go from the bipolar cells to the ganglion cells. And now these ganglion cells are collecting like a bunch of bipolar cells synapse onto a few ganglion cells. They're kind of collecting all of that information, that regional information. And that's when they send it to the optic nerve. Because this is kind of the key where we have to get this information that these eyeballs into the brain. And therefore, there must be a nerve that's sending it from the eyeballs to the brain, and that's what we know as the optic nerve. This optic nerve is collecting all of this information from all of these ganglion cells, because when like photoreceptor, bipolar, ganglion, now to the optic nerve, collecting all that information, and it will leave the eye. If you've ever done a dissection of an eye in lab, it's not just you don't just see the ball of the eye. There's usually something sticking out the back. That's your optic nerve. That's the optic nerve that will be going out collecting all of that visual information and sending it to the brain, and we'll talk about what goes there. And it's actually very interesting because it has to leave the brain. There's actually this one area of the eye that has no rods or cones. This is what's known as the blind spot, right? The place where your optic nerve leaves the eyeball, right? There's no rods or cones there. So if you can always like test for your like blind spot, like I know when I had a textbook, like it's like, oh, hey, keep on looking until you can't see the dot anymore. You just identified your blind spot. So if you're ever curious, you can just look up like blind spot exercise and you can kind of look it up. I'll leave that for homework to gotta go find your blind spot. But you have a blind spot and it's because there's an area of your eye where the optic nerve is leaving your eyeball. That's where there's no rods or cones, so therefore no vision.
Yeah, it's so cool.
Mhm.
It's so cool. And
Mhm.
because you keep bringing up evolutionary biology, right? Like, why the heck would you ever program a blind spot? It's like, okay, well, if we didn't have light leaving the eye to go to the brain, you wouldn't see anything. Okay, so it's a trade-off. Okay, I need to be able to see. So
Mhm. Mhm.
the fact is, I need an optic nerve, right? And you're
Mhm.
right. If you've done dissections, whether it's an anatomy lab or I mean, heck, I think I originally dissected an eye in like middle school or high school bio. Right? It's you see the actual ball itself, and it's like almost like a stem coming off of it, right? Now, this optic nerve, like you said, is carrying this visual information, and it's going to go to the brain, but it's actually got a pretty complicated way of getting there. And I think that the complications actually make sense with evolutionary biology. So
Of
thank you
course they
for
do.
bringing it up, Mike. Right? Oftentimes, what we would expect is like, oh, okay, we know that the part of the brain that is receiving a signal, it's actually coming from the opposite part of the body and vice versa, right? So it's like let's say, the motor impulses that are moving my left hand come from my right motor cortex. Okay, or if I poke my left hand, that's being sensed by my right somatosensory cortex, right? It's all contralateral, it's opposite sides. You would expect the information coming from the left eye to go to the right occipital lobe, right? And vice versa, right eye to the left, right? Contralateral. Makes sense. But
Hmm.
the reality is my left eye, right? If you close your eye, go ahead, do this. Close one of your eyes. How far can you see? Right? Like
I see
most of
both
us,
directions.
Yeah, you could see I mean, you could see a pretty wide field of view.
I see my
Okay,
nose
like
too. That's cool.
even without one, you can maybe not see, let's say if I close my right eye, maybe I couldn't see like my far right visual field, but I could still see a lot,
Hmm.
right? The reality is each one of your eyes is picking up light coming from both visual fields. And that's like if you drew a line from the center of your forehead or your nose out straight, okay, the left side of that is your left visual field, right is on the other side. So the reason for this complicated system we're going to talk about with the optic nerves crossing over at what we call the optic chiasm and then traveling through tracks to the occipital lobe or the cortex. The reason that they do that is actually to sort such that the right occipital lobe is not picking up everything that the left eye sees, but all the information in the left visual field and vice versa. The left sees everything in the right visual field. That's the goal. Okay, that being said, we do need to understand how these sources of information are crossing over. Okay, and this is where it's nice to have diagrams, right? But if you took both retinas, okay, you could divide it in half straight down the middle where your pupil is. Okay, and you can get a nasal side, right? That would be towards the midline, right? Or towards your nose, really the inside portion of your eye, versus the temporal, right? Which is out towards, let's say, your ears, okay, or your temporal lobe. It's on the sides, right? Um, Now, when we think of which visual field you're picking up on, this is where I think it gets a little bit more confusing. I encourage you to take a look at an actual diagram for this to be able to see what's going on, right? And how these different regions of the retina are seeing different visual fields. But I just like to imagine, right, like if something is on my right visual field, and I'm thinking of my how that's coming into my left eye, right? Right visual field, it's going to come at an angle, and it would be really hard for it to strike the nasal side of my retina because it would kind of have to pass through my nose, right? Like if it's all the way on the other side of my body, it's probably going to go to the outside of my eye, right? That's how I like to think about it. So like my left eye, the part of my left eye that's perceiving or receiving information from my right visual field, it's going to probably come over to the temporal side, right? And that's what we want, right? That's what we want. So it is the nasal retinal fibers that are going to cross. Okay, so at that optic chiasm, we are essentially sorting, instead of information, which eye are you coming from? We're sorting which visual field are you perceiving information from. Okay, we
Hmm.
want to sort such that when you leave the optic chiasm and go into the optic tracts and you're heading out essentially to the occipital lobe, one tract is going to have information from the left visual field, one tract is going to have it from the right visual field.
Yeah.
Again, I know it's confusing. Again,
Mhm.
I know, Mike, you're like, I wouldn't have designed it this way. I wouldn't want to design it this way. However, I do understand why it's important
Mhm.
because if I'm, trying to evade a predator, oh, I see the predator with my left eye. That doesn't help me, right? Like,
Mhm.
I have no idea where I should run, where I should fight, right? Like, that's not helpful. I need to know, oh, the predator is on my left side or on my right side so that I can run in the opposite direction
Right,
or
right.
towards it if I'm feeling a little frisky and try and fight
If
it,
you're,
right?
if
But
yeah, maybe it's not flight, maybe it's fight.
it's
Maybe we're,
fight.
maybe
Either way, it's very important in an evolutionary sense, and even in our world today, that we can localize where sight is coming from, or where visual information is coming from. So
right.
super important. And really, we're almost there, right? Optic tracts. You stop at what's called the lateral geniculate nucleus, which is a part of the thalamus, which is that sensory relay station of the brain, and then you go out to the cortex.
Right.
And that's almost the whole story. Now we're in the brain,
Hmm.
and then a whole nother layer of things happen.
There's another. So yeah, you're like, oh, we're over there. Actually, there's one more. But yeah, no, I love, like, I was always confused when I was studying with the eyes, especially, like I was always, oh yeah, right eye goes to left brain. Oh, left eye goes right. But yeah, it's
Hmm.
not by eye, it's by visual field. Right
Yes.
visual field information goes to the left side of the brain.
Yes.
Left visual field goes to the right side. But because each eye sees both, they have to do a little bit of sorting. So yeah, I think you put that really, really well. That it's like, yeah, it's not by eye, it's by field. 100%.
Yes.
But now we have to, like, okay, it's still a signal though, right? That's the thing. It's like still just electrical signal. There's been no sensation. Like we have this, but there's been no perception of it. Our brain has not processed this information because all the information that you're really seeing, like I'm seeing a face and you're seeing a face. Like you're seeing my face, but you're not, that's not actually what you're seeing. Really, you're seeing like edges and lines and details, but those details need to be coalesced, need to be combined, and then you can see an image. That's where the next phase comes in, where this is where we get to the occipital lobe, where the occipital lobe, again, I don't know who designed this, but it's in the back of the brain. As far as like, you went to the point furthest away from the eyes, right? You would think like the eyes are on the front of the head. Oh, where'd be the occipital lobe? Oh, pretty close. No, it's on the opposite side of the head. So the occipital lobe, it has to travel all the way to the back. And I'm sure there's a reason about this. I'm sure there is. I'm sure Molly's like, Mike, this is the reason, XYZ, evolutionary biology wise. But for me, I'm like, you know what? As a human, I would think it'd be close. But anyway, I like to remember occipital lobe is on the exact opposite side of the brain. Eyes in the front of the brain, at the very front of your head, occipital lobe at the back. So we've our optic tracts have synapses on the LGN, on the lateral geniculate nucleus. We have these optic radiations, and now they've been set at the occipital lobe, specifically an area of the occipital lobe known as the primary visual cortex. Now, that's, again, the name simply tells you most of what you need to know, is that this is where our visual information is first going to go. It's going to first synapse at the primary visual cortex, and it's kind of like mapped out on this cortex. So what I mean is like information that's like next to each other in your visual field is going to be next to each other on the occipital lobe. It's kind of what we talked about before. Remember when we discussed like the motor cortex kind of deal, where we had the the homunculus, right? Where you could like
Oh
map
God, don't
out,
bring that
yeah,
up.
that freaky thing. Anyway, the homunculus, where it's like you can map out all the parts of the body on the motor cortex. Same idea with the primary visual cortex, but like it's instead of it's like motor areas and sensory areas of your whole body, now it's like, oh yeah, things that are next to each other in that you saw are going to be mapped next to each other on this visual cortex and deal. So they're kind of mapped out similarly. And when it's kind of like mapping out on that cortex, what we're actually sensing is a number of different details about what you're seeing. Again, you're not just seeing faces, you're seeing kind of four processes. You are detecting motion, color, depth, and you are having a little bit of object recognition. And when you're doing all those processes are going on in that cortex to kind of see like, hey, I'm seeing lines and edges. What does that mean? Right? When we're perceiving, we combine meaning with what we're seeing. Like, oh, I saw this thing go, I saw a detail here, and then it was here 3 seconds later. Oh, that means it's moving. Oh, this sort of wavelength was activated. Oh, that means it's orange. Oh, I actually know that face. I see her every single week. Oh, that's Molly, right? Like my eye didn't pick up these things. It was when I took that visual information and it went to my occipital lobe and I was able to combine it with meaning. And that's when we've actually perceived. And it's kind of like this idea that vision is not the same thing as perception. Vision is the sensation. Vision is like, oh, I like my things have been activated. Like my cones, my rods have been activated. But it's when I add meaning to those things, now I've perceived those things. And that's kind of like the kind of the role of the brain is to add that meaning in order to have perception. And what's also very cool on that mapping of the brain is about how it's mapped on the brain in terms of a little bit of review of last episode, where when we had a real image, like our lens is a converging lens, which would create a real image. But what was that detail about real images we talked about kind of last episode, Molly?
Let me think about it.
Yeah.
I remember there were some trends,
Hmm.
right? And they had something to do with actually the electromagnetic spectrum, at least how I remember it, right? We
Hmm.
had real and virtual images, right? R and V. And I remember that there was a little mnemonic, right? IR, UV, infrared,
Hmm.
ultraviolet, right?
Hmm.
Real images are always inverted,
Inverted, right?
whereas virtual images are always upright. Oh,
Right.
wait, hold
And
on. Wait, wait,
wait a
wait.
minute.
Are you saying that when I see that the ceiling is up, is that mean it's down? Hold on. What is going on with that?
In your brain, yes. When you first picked up that information, like when it was mapped on the retina, because it was mapped on the retina, it was upside down. All that, like when I saw the ceiling, it was mapped on the bottom of my retina, and when it was mapped on my brain, on my visual cortex, it was inverted. And our brain, in the process of perception, needs to uninvert it essentially, which
Whoa.
is so cool, right? Where our brain, again, this is the whole idea of how involved the brain is in our sight. It needs to essentially take the entire world and flip it upside down, and that's the only reason that you can see properly. If this was not working correctly, you would be seeing everything upside down, which would be the most vertigo-inducing situation like ever, which is wild. But something that's so cool because like and when you combine like I remember like when you learn these things individually, like, oh yeah, visual stuff makes sense. Oh, lens stuff makes sense. But wait a minute, then you're like, wait a minute, upside that this should be upside down. Why aren't I seeing the world upside down? Because it's the brain essentially fixing what, kind of like your lens did to that light.
Yeah. It's so cool. The brain
Mhm.
is such a fascinating organ, which is why, of course, we're spending time talking about it. And we've gone on a couple little super fun tangents, right? About different types of physics concepts and how that impacts different sensory organs, etc. Because it's all interrelated. And the brain really is so critical in helping us evaluate how to respond to different signals in our world, right? So I love that you bring that up, that we really technically sense the world upside down, and yet we are able to navigate the world sensically in a way that makes sense because of the actions of the brain. So I love that. And I feel like that really kind of wraps up what we learn in the visual pathway, right? Again, we really started with just light waves traveling through the air, right? They have to physically cross from the light source to you, right? And to your eye. It's going to pass through the cornea and the lens, which is going to cause refraction and bending. Hopefully, right? If you're like Mike and you have 20/20 vision, hopefully that light is focused directly on the retina, upside down, but directly on it,
Hmm.
which would allow proper, clear vision. If not, if it's too far before, right, too early, it's focusing with myopia, right? Or if it's too far back with hyperopia, you might need an external lens. But once we get light perfectly focused on the retina, however it gets there, right, we have a whole transduction cascade in the retina itself. That visual information is going to be traveling through the optic nerve, past that optic chiasm, into optic tracts, through that lateral geniculate nucleus, and to the occipital lobe for actual perception. And of course, once you understand what's going on in your environment, you can then make decisions on how you would like to move throughout your environment, right? Or, you might decide to say something or, do something. Who knows, right? This information is incredibly critical as creatures or as a species that are incredibly visual or visually dependent. So pretty awesome. I love that we get to go through this. And in our next episode, we get to actually really zoom in on something that is involved in a lot of what we've talked about with senses so far, and actually very, very important in other contexts, which is signaling cascades. And we'll look specifically about G protein coupled receptors. But like, how does this work? How do you have signaling? How can one cell tell another cell to do something else,
Hmm.
essentially? So
Right.
I'm excited to talk about that too.
It's such a cool content area because when we communicate, it's like, oh, just use your words, dude. Like just like speak. But cells can't speak, or can they, in their own kind of special little way? And so this is kind of the whole idea of signaling cascades. How do cells communicate? How do they convey, hey, you should do this without speaking? Question mark. We'll talk about that next episode. Super excited to talk about that. Until then. Hope everyone has a great rest of their day, evening, night, train ride, bus ride, wherever we find you. Until the next episode, happy studying, everybody.
Bye.